Hello everyone, in this video lecture we will be discussing some MCQs from the chapter Immunity from AS Level Biology 9700. Let's start. Question number 38. What are the functions of T lymphocytes during an immune response? So if we talk about T lymphocytes are of two types. What are those? One is T helper lymphocyte and the other one is T killer lymphocyte, right? So there are two types of T lymphocytes, T helper lymphocytes and T killer lymphocytes. So we have to state the functions of T lymphocytes during an immune response. Let's read the first statement. Destroy infected body cells. This is the function of T killer lymphocytes. What T killer lymphocytes do, that they destroy host cells which are infected by a virus, and T killer lymphocytes also destroy cancer cells. Differentiate into memory cells, both T helper lymphocytes and T killer lymphocytes. Differentiate or specialize into memory cells for secondary immune response, right? Secrete antibodies. So always remember this point, that antibodies are secreted by plasma cells, and plasma cells are formed by the differentiation of B lymphocytes. So T lymphocytes have nothing to do with secretion of antibodies. The answer is A, 1 and 2. Let's move on to MCQ number 37. Rabies is a viral disease which can be spread to humans by a bite from an infected animal. One method of treatment is to inject the patient with antibodies specific to the rabies virus. Which statement about this treatment are correct? Okay, so they are talking about rabies, which is a viral infection. It's caused by rabies virus and it spreads to humans when some infected animal like dog or like bats, or we can say raccoons, when they bite humans. So this virus can be transmitted from the infected animal to humans. So how do we treat rabies? We treat rabies by injecting antibodies which are against rabies viruses, right? So remember this point, that whenever you are injecting antibodies into a person to provide protection against a pathogen or an infection, so that is called artificial passive immunity. We call it artificial because we are injecting antibodies, right? So that's why artificial. It's not a natural process. Then because we are injecting antibodies, so this is passive immunity. What is meant by immunity? Immunity means protection against a particular infection or a disease or a pathogen. That is immunity. And immunity can be active or passive. So when we inject antibodies, immunity is passive. When we inject antigen, immunity is active. And the reason is that when you inject antigen, immune response occurs. And because immune response is occurring, this is an active process. So when you inject an antigen into a human, what happens? That immune response is initiated, B lymphocytes are activated, T lymphocytes are activated, B lymphocytes specialize into plasma cells secrete antibodies. So that is an active process, and this is known as active immunity. But when you're injecting antibodies, so immune response is not initiated. The protection is simply provided by the antibodies that you're injecting. So that is passive immunity, okay? And because you're injecting antibodies into a person, that is artificial process, because you are injecting. The person is not getting antibodies from a natural source, all right? So which statements about this treatment are correct? The patient will have natural passive immunity to rabies? No. The patient will have artificial passive immunity to rabies, not natural, okay? So statement 1 is wrong. The injected antibodies will be broken down by the patient. Yes, this is true. When you inject antibodies into a person, antibodies will eventually be broken down by the liver. The patient's memory cells will be able to produce this antibody more rapidly in the future. Remember this point, that when you inject antibody, immune response, when you inject antibodies, immune response is not initiated. Memory cells are not produced. So memory cells will not be available, and they will not be available to produce antibodies in the future, right? So this is wrong. The immunity provided will be of short duration. This is true because you have just injected antibodies which will be broken down in a month or two. So the immunity provided will be short. But if you would have injected a vaccine, for example, if you would have injected a rabies vaccine, which would contain a weakened rabies virus, so that would cause an immune response, right? So whenever you inject an antigen or a pathogen, right, immune response is initiated, B lymphocytes and T lymphocytes are activated, and memory cells are produced. So memory cells act as immunological memory. So when the pathogen comes, or when you get infected with a pathogen, so memory cells are there which rapidly produce antibodies, and you're protected from future infections. But when you inject antibodies, immune response is not initiated, memory cells are not made, so immunity is only for short term. Let's move on to MCQ number 36. What describes a function of a macrophage? Okay, macrophages, always remember this point, they're found in tissues. And they secrete cytokines in response to an infection. So guys, what is meant by cytokine? Cytokine is a chemical messenger molecule which is used by the white blood cells to communicate with each other. So white blood cells secrete cytokines, all the white blood cells secrete cytokines to communicate with each other, right? So macrophages are the white blood cells which are always found in tissues. All right? And they can secrete cytokines in response to infection, right? If you look at other options, they can leave the blood. So if we talk about leaving the blood, macrophages don't leave the blood. Actually, monocytes leave the blood. So what is the difference between monocyte and macrophages? Monocytes are the precursors of macrophages. What is meant by precursor? So macrophage is initially a monocyte, and then monocyte becomes a macrophage when it goes into the tissue. So monocytes actually leave the blood and go into the tissue and become macrophages. So always remember this point, that macrophages are always found in tissues. When the same cell is leaving the blood, it's not called macrophage, it's called monocyte. So this is wrong, right? Circulate in the blood. All of these points are wrong. Okay. Let's move on to MCQ number 35. A person's blood group is determined by the antigens present on the red blood cells. Most people have antibodies in their blood plasma even if they have never received a blood transfusion. It is these antibodies in the plasma of a person who receives the blood which makes some blood transfusions unsafe. So the information over here says that the blood group is determined by the antigens present on the red blood cells. So let me give you an idea. So for example, these are the red blood cells. This is the first red blood cell, this is the second one, this is third one, and this is fourth one. So blood group is determined by the antigens on the surface of the red blood cells. So for example, if red blood cell has A antigen on its surface, the blood group is A. If red blood cell has B antigen on its surface, the blood group is B. If red blood cell has both A antigen and B antigen on its surface, the blood group is AB. And if red blood cell does not have any antigen on the surface, the blood group is O, right? This is the concept. So if a person has blood group A, the red blood cells of that person will only have A antigen on the surface. If a person has a blood group B, so the red blood cells of that person will contain B antigens on the surface. If a person has blood group AB, then there will be both A and B antigens on the surface of red blood cells. If a person has blood group O, there will be no antigens on the surface of red blood cells, right? Okay, so over here we can see the table. So if a person has blood group A, this means that antigen on the red blood cells is A. However, the same patient will have antibodies to B. So let me give you an idea. This person has blood group A, right? Because of A antigen on the surface of red blood cell, but this person will have an antibody against B antigen, right? If a person has a blood group A with A antigen on the surface, the same person will have antibodies against B and not A, right? Because for this person, antigen B is foreign, so antibodies will be against B. If a person has a blood group B, so on the surface of red blood cells, there will be B antigens, and the same person will have antibodies against A antigen. If a person has blood group AB, so there will be B and A antigens on the surface and there will be no antibodies. Because antibodies are only formed against foreign antigens. Always remember this point. So if a person has blood group AB, so both A and B antigens are self antigens. So there will be no antibodies against A or B, right? But if a person has blood group B, so for this person, antigen A is foreign, so there will be antibody against A, right? If a person has blood group A, so for this person, antigen A is self and antigen B is foreign. So this person will have antibody against B. If a person has blood group O, there are no antigens, which means that for this person, both antigens A and B are foreign, so this person will have antibodies against A and antibody against B. All right, this is the rule. So you can see that in the table as well. If a person has blood group A, which means that this person has A as a self antigen and B as a foreign antigen, so antibody will be against B. If a person has blood group B, so B will be self antigen, A will be non-self, and there will be antibody against A. If a person has AB, so both A and B are self antigens, right? No foreign antigens. So there will be no antibodies to A and B because both antigens are self. If we talk about blood group O, so for the person with blood group O, the person does not have A or B antigens. So both of the antigens are foreign. So this patient or this person will have antibodies against both A and B. Now the question is, which are the blood groups of people who can safely receive blood from a person who has blood group A? So they are asking that which person can receive a blood group A blood safely. For example, if I were to receive a blood transfusion from a person with blood group A, right? Then I should not have any antibody against A antigen. Are you guys getting my point? Like if I have to receive blood transfusion from a person with blood group A, then I should not have antibody against A. Because in that case, if I have the antibody, what will happen? There will be tissue rejection. So if you transfuse a blood with blood group A in me, and if I have antibodies against antigen A, so that blood will be destroyed or rejected, and I will get severe reaction, right? So whenever you are transfusing blood, you need to make sure that the person that is receiving the blood does not have antibody against the red blood cells that you are transfusing. So for example, if you're transfusing a blood with blood group A, then the recipient should not have antibody against A antigen. So which are the blood groups of people who can safely receive blood from a person who has blood group A? The answer should be A and AB, because we can see in the table, if we talk about A and AB, so these two blood groups have no antibody against A. So they can safely receive blood from a person who has blood group A. Let's move on to MCQ number 34. An antiserum to a snake toxin can be obtained by injecting the toxin into a horse. The antiserum is made from blood plasma taken from the horse a few weeks later. The antiserum is injected into a person who has been bitten by the same species of snake. Which type of immunity occurs as a result of using this antiserum? So, guys, what is the story in this question? So, they are saying that we can inject a snake toxin into a horse, and horse can then make antibodies against this toxin. Then what we can do is that we can take blood plasma from that horse, which will contain the antibodies. So basically antiserum contains what? Antiserum is basically the blood plasma taken from the horse, which will contain antibodies against what? Against snake toxin. Are you guys getting my point? What are we doing is that we are injecting a snake toxin into a horse. Obviously, horse will respond to that toxin in such a way that immune response will be initiated, and B lymphocytes will be activated, and B lymphocytes will specialize into plasma cells. Plasma cells will secrete antibody into the blood plasma. So there will be antibodies in the blood plasma against this toxin. And if we obtain that blood plasma, which they are calling as antiserum, that antiserum will contain antibodies against snake toxin. Now they are saying that this antiserum, which was obtained from the horse, is injected into a person who has been bitten by the same species of snake. What type of immunity occurs as a result of using this antiserum? So if you're injecting antiserum into a human, you are basically injecting antibodies. So whenever you inject antibodies to provide immunity, that is artificial passive. Why artificial? Because you're injecting. And why passive? Because you're giving the patient antibodies and not an antigen. Right? So the answer is B.